This is David Johnson with my first video on blood cells and blood plasma proteins. Here's my email address and other information, my office phone number. Our objectives for this section are to list the major blood cell types and their functions, describe how blood plasma differs from serum, define acute phase reactants and give examples, identify major blood proteins with their electrophoretic peak, list functions of plasma proteins and organ of synthesis, tell why alpha-1 antitrypsin A1AT deficiency results in emphysema. This is a picture showing the various blood cells. You can see at the very top is the hematopoietic stem cell, and that divides into various others. There's a common lymphoid progenitor, and that common lymphoid progenitor gives rise to B cells and T cells, which are lymphocytes, and then for natural killer cells, which are also part of that, the adaptive immunity or cell-mediated immunity. And plasma cells make immunoglobulins, make the IgGs that we'll talk about later. The next section is the myeloid progenitors, and myeloid cells become granulocytes such as neutrophils, basophils, and eosinophils. And the neutrophil attacks bacteria. The basophil is a blood cell very much like the mast cell, but it's soluble in the blood. And eosinophils also act on parasites. Then also in the myeloid section are the monocytes. And they go in, the monocyte becomes a dendritic cell or a macrophage, and these are antigen presenting cells, and they present these antigens they attack bacteria and other things and present them to the plasma, to the T cells, so that they can initiate the B cells to make new antibodies. The mast cell is a tissue cell, and it contains IgE bound to its surface, and it also has these granule-rich components. These granules that are rich in histamine, and so it's a very histamine-producing cell. And so we take antihistamines to keep the mast cells from degranulating, because when the mast cells degranulate, they release histamine, and that causes an edema, and we get runny eyes and runny noses. The erythropoietic cells become megakaryocytes and erythroblasts, and the erythroblasts then become reticulocytes, and the reticulocytes become erythrocytes, which are the red blood cells that carry oxygen to our tissues, from the lungs to our tissues. Very important. We'll talk more about the erythrocytes. They are the major blood cell. The platelets, the megakaryocyte breaks up into little pieces we call platelets, and those are also called thrombocytes, and they last about 10 days. And they are very involved in blood clotting. We'll talk about those as well. And the erythrocyte lives for about 120 days, and that makes it a very useful cell for various clinical studies as well. Now, blood plasma has two major components. It has cells, and it has this liquid we call plasma, and that straw-colored liquid has salts and vitamins and urea, amino acids and proteins and fats in it. And then if you let blood settle, you'll see platelets, white blood cells and platelets at the top, and then red blood cells down below. And this is sometimes called the buffy coat, right in here in the middle, the white coat right between the red blood cells and the plasma. Plasma liquid is what you get from your centrifuge blood. And to get it separated, you need to add an anticoagulant to keep the blood from clotting, such as EDTA, which is a lavender top, citrate a blue top, and heparin a green top. You won't be asked that on the test, but these are all anticoagulants that slow the process, and you'll learn more about them as time goes on. Serum is the liquid from coagulated blood, and the red blood cells make up about 45% of the blood, and so they're the major component down there. And in serum, fibrinogen gets converted to fibrin, and fibrin's insoluble. That's what forms the clot. Prothrombin is the enzyme that activates, that converts fibrinogen into fibrin, and so prothrombin gets activated to thrombin, which is an active serine protease enzyme. Now, red blood cells are pictured here. You can see the red blood cell in pink. There's a lymphocyte in blue and platelets in yellow in relative size of these cells. So you can see what sort of they look like. The erythrocytes contain hemoglobin and transport oxygen. They live an average 120 days. And you can see their sort of disc-like cells with a concave middle, and that allows the greater surface area so oxygen can bind more readily. The red blood cells lack nuclei and mitochondria, making them biconcave and very flexible so they can squeeze through capillaries. That's a very important function. That's one of the reasons they don't have a nucleus. Reticulocytes are the precursors of red blood cells. So here is normal serum electrophoresis. If you take serum and do electrophoresis on it, you'll see this pattern. You can see the pattern across the bottom, and if you scan that, you can get an approximate contribution of each component to that. You can see that prealbumin is the major one. Albumin accounts for about 50% of the blood plasma proteins, and there's an area called prealbumin. You don't see a peak there, but there are some proteins in there called prealbumins. Then you have the alpha 1, alpha 2, beta, and gamma region. In the gamma region, we have the immunoglobulins, IgG, IgA, and IgM. And they have a broad peak because they're polyclonal in normal people. That means they recognize a whole range of different antigens. That's why they're called polyclonal, because they come from different lymphocytes. So you'll have a multiple host of B cells that are making IgG, another bunch that are making IgA, and another bunch that are making IgM. Acute phase proteins. Several plasma protein concentrations increase in response to inflammation and or infection. C-reactive protein is an example. It binds to phosphocholine on microorganisms and may enhance phagocytosis by macrophage. Orosomucoid, or also known as alpha-1 acid glycoproteins. Plasma levels increase in pregnancy, burns, certain drugs, and certain diseases, particularly HIV. It's a carrier of basic positively charged and lymphophilic compounds and drugs. Alpha-1 antitrypsin, which we'll talk about more later, increases to inhibit elastase release from neutrophils due to inflammation. Albumin is the major component of blood plasma. It's about 50% of the plasma proteins, and it's responsible for osmotic pressure. And it's used to treat shock. Quite often, it was used back in World War II. The first time pure plasma albumin was used you could add sterile water to it and give it to injured soldiers on the battlefield and expand their blood plasma so they did not go into shock. Analbuminemia is very rare. That's no albumin, and I've only heard about one case of that ever occurring. Hypoalbuminemia, hepatic damage due to reduced synthesis, or syndromes such as loss in the urine. Albumin is also a negative acute phase reactance. It decreases when others increase, and that maintains the osmotic pressure. Albumin binds and transports bilirubin. We'll talk more about that later when we talk about heme biosynthesis and degradation. It's because bilirubin is a heme breakdown product or degradation product, and heme is the chromophoric group that's in red blood cells that makes them red. It contains a lot of aspartic acid and glutamic acid and has an isoelectric point of about 5. It's metal ion binding because of that. Metal ions are positive charged, albumin's negatively charged, and it also has, since it binds bilirubin, that's a hydrophobic compound, it binds under hydrophobic things such as drugs. And in the alpha-1 and alpha-2 regions, in alpha-1, we have alpha-1 antitrypsin. It's a serpin. It's a serine proteinase inhibitor. It's also called alpha-1 proteinase inhibitor, and it specifically inhibits human neutrophil elastase. So its target is to make sure that neutrophil elastase, when the white blood cells die, or neutrophils die, and they don't live very long, maybe about a day, then they release their enzyme, and you wouldn't want that degrading away your tissues and attacking stuff. So you don't want to keep this protease under control. And then in the alpha-2 region, we have ceruloplasmin, which is a copper transporter. We have GC globulin, which is a vitamin D carrier. We have haptoglobin, which binds hemoglobin 1-to-1. We have prothrombin, that's a zymogen of thrombin, which I've mentioned previously. We have antithrombin 3, which is another serpin, and it inhibits thrombin and some other clotting factors, but it has to be activated by heparin, or it's activated by things like heparin, like heparan, that's found on the surface of our endothelial cells. Now, alpha-1 antitrypsin deficiency came to the fore in 1963 through the work of a biochemist, C.B. Laurel, and a physician named Eriksson. And they were working in Malmo, Sweden. And Eriksson had a number of patients that were young and had emphysema, and he wanted to know why. And Laurel did their serum electrophoresis on them and found that they were missing the alpha-1 peak. And so this resulted in a hypothesis, since we knew that back then it was called, this was the major trypsin inhibitor blood. We didn't know it inhibited elastase back then. So they came up with the hypothesis that proteases can cause emphysema. And people did various studies in animals showing that if you put elastase into their lungs, you can create an emphysemus-like environment. However, remember that 80 to 90% of emphysema is due to cigarette smoking. Probably 10 to 20% is due to other environmental causes. The alpha-1 antitrypsin deficiency is only about 1%, but it's fairly prevalent in Northern Europeans. Neutrophils and macrophage produce elastases, and smoking produces oxidants, and those oxidants will inactivate alpha-1 proteinase inhibitor, and that will allow the elastases of the neutrophils and the macrophage to start damaging the tissues in the lung. And that's all you really need to know about that. Smoking is thought to oxidize alpha-1 AT in the lung, resulting in a deficiency of active inhibitor in the lungs. And there's the methionine structure. And then prolastin made by Bayer is just alpha-1 antitrypsin isolated from blood plasma, and it's used to treat the genetic deficiency of alpha-1 antitrypsin. So if you oxidize that methionine, it has one oxygen on it, and that oxygen reduces the ability of the alpha-1 antitrypsin to inhibit elastase. It doesn't fit in the binding pocket quite as well, and that's called methionine sulfoxide. And I discovered the presence of methionine in alpha-1 antitrypsin, and I discovered that the oxidation effect. So here is a model showing elastase. This work was done by James Huntington, Randy Reed, and Robin Carroll at Cambridge back in 2000, and they found that elastase would cleave methionine, and in that process do a rearrangement. The alpha-1 antitrypsin rearranges, and in the structure it results in inhibiting elastase by disrupting its structure. There are other serpins mentioned previously, antithrombin 3. There's also another one called alpha-1 antiplasmin, and there's one called alpha-1 antichymotrypsin. So all of these work very similarly. So here's the mechanism by which this inhibits. You can see the elastase coming down to hit the methionine, cleave at the methionine inhibitory site. And once it does that, then you get a rearrangement, and you get the inactive protease down at the bottom, which is no longer functional. So in the beta region, we have hemopexin that binds heme 1-to-1. We have transferrin that binds iron, 2 moles of iron, Fe plus 3 per mole. And we have 3 iron conservation proteins: haptoglobin, which binds hemoglobin, hemopexin, which binds heme, and transferrin, which binds iron. Iron is kind of hard to get into our bodies, and we don't want to lose it. We really need it because we're constantly making red blood cells and cytochromes. We'll talk about later. There are enzymes in the mitochondria that are very important, but you want to keep an adequate amount of iron, and so we don't want to lose that iron out in the urine, so we have all these proteins to keep it. Iron conservation proteins, haptoglobin 1-to-1, that's its concentration. You don't need to memorize those. Hemopexin And transferrin delivers iron for to cells for storage. And then ferritin is an intracellular protein that stores iron. Blood levels can be very high, are very low, only about 30 to 300 nanograms, because it's generally intracellular. Diagnostic for anemia, low ferritin leukocytemia, 424 units of subunits, and it binds 4,500 irons each. So it binds a lot of irons. Do not memorize these concentrations. You do not need to know them. You can always look them up. Then the Phe region, we have fibrinogen. And I've told you previously that fibrinogen gets converted to fibrin, which are polymers that form blood clots. Thrombin makes two little cuts in this big molecule called fibrinogen that has 2 alpha chains, 2 beta chains, and 2 gamma chains. And it cuts only the alpha chains and the beta chains. And it cuts them very close to the N-terminus, cuts both of them. And then that changes the charge, which allows this whole these molecules to polymerize, and that results in a blood clot. And remember, don't smoke and be careful what you drink. Thank you, and that's all for right now.